Hello and welcome to the eOrganic webinar on Organic Farming Systems Research at the University of Nebraska. My name is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. You can find all of our published articles, videos, and our many upcoming and recorded webinars on organic farming and research topics on our website at extension.org/organic_production. So before we begin, I'd like to briefly introduce our speakers. Liz Sarno is an extension educator for organic farming systems in Nebraska. She coordinates the Organic Working Group team, and she also owns and operates an organic grass-finished Devon cattle farm in eastern Nebraska. Charles Shapiro is a professor of agronomy and horticulture. He has been researching soil fertility issues for 30 years, and he has managed organic research land at the Heskel Agricultural Laboratory since 2005. Richard Little has 15 years of experience in hard winter wheat breeding in the Northern Plains, 10 years in South Dakota, and 5 years as the research coordinator for the organic wheat breeding program at UNL. His focus on grain quality testing in South Dakota evolved into a hobby of whole wheat sourdough baking and a current series project involving artisan bakers. Vicki Schlegel is an associate professor in food science and technology at UNL. Her research includes characterizing the small molecules in natural systems to understand the effects of farming practices on plant secondary metabolites and how they affect human health. Jim Brandle is a professor of forestry in the School of Natural Resources. He will be discussing the Healthy Farm Index as a way of measuring the impact of organic practices on biodiversity.
I'm Liz Sarno, Extension Educator and Project Coordinator. I'll give you a short overview of the Organic Research Program at the University of Nebraska. Organic research has been conducted at the University of Nebraska since 1976. Ag researchers thought it was important to establish a long-term experiment that evaluated a crop rotation under organic and conventional management. The program that I coordinate now started in 2006 with a project called Improving Organic Farming Systems Across Nebraska's agro-eco regions. It was a 4-year project funded with a USDA grant. Along with the grant, we developed an organic working group, which is composed of a team of soil scientists, entomologists, weed scientists, and economists, and extension educators, organic technicians, graduate students, and others. One of the goals set in the first grant was to establish the university's first certified NOP organic farm ground focused on organic research projects in 3 of Nebraska's 4 ecoregions. UNL received funding for a second USDA OREI grant, Improving Organic Farming Systems and assessing their environmental impacts across agro-ecoregions. You will hear about these objectives of this grant from my colleagues today in this PowerPoint. Some of our current organic research areas are nutrient management, weed management, cover crops, organic small grains breeding and systems development, antioxidant production in organic small grains, biodiversity, conservation on working organic farms, and the Healthy Farm Index assessment of biodiversity indicators and land use patterns. As you can see, it's about 450 miles across Nebraska on Interstate 80. It's a rather big state. Nebraska has a diverse ecological and cultural characteristics. The average rainfall ranges from 13 inches in the west to 36 inches in the eastern part of the state. Differing lengths of growing seasons, altitudes, and soils that are valuable for screening cultivars and developing crops for adaptation to organic management under a variety of conditions. Research is conducted on certified land adhering to the National Organic Program regulations, and I think that's an important point that we always follow the NOP, the National Organic Regulations, using accepted cultural practices and crop rotations for the varying conditions. This helps us to provide organic crop production solutions for farmers in their ecoregions. Oops, I hope I didn't. Yeah, there we go. I'm going to take you to our first research center, the ARDC, which is our Agricultural Research Development Center out at Mead. We have 45 acres that are protected by mature shelters, belts, and 7 acres that are part of an ecological study experimenting with cover crop mixtures. They both are following the rotational practices in the area. So they have corn and soybeans, winter wheat. On the shelter belt area, we have a mixture of clovers that are being interplanted with the winter wheat to try to improve fertility. Heskel Ag Lab up by Concord has 33 acres of NOP certified ground. We are doing experiments with cover crop mixtures, oats and clovers, alfalfas. The alfalfas are fed to the livestock that are on the farm and are also used as green manure in the rotation. One important part is to destroy the alfalfa. We are also looking at different types of equipment. So we found it's best to use a noble or undercutter blade to destroy the crop, so we're not taking up a lot of the moisture. That's pretty valuable in Nebraska. And we also received a Sears Trust grant where we're going to be looking at various methods of crimping certain cover crops and trying to directly plant into that. South Central Ag Lab is in Clay Center. We have over 21 acres in certified NOP ground. This is the only farm that we are able to have irrigated strips. And what we did with this farm is we set it up in strips, so that way we have better weed control for our experiments. And we also have popcorn in this particular plot because that tends to do well under irrigation, and it's definitely a good cash crop for the farmers trying to go organic here. And the furthest away out west is our High Plains Agricultural Laboratory, where we have 76 acres of NOP certified organic ground. This has a rotation of 2 3-year crops, rotations stacked on top of each other, so it gives us a 6-year rotation. The rotation is winter wheat, proso millet, green manure fallow, winter wheat, sunflowers, and fallow with composted manure. And fallow is every 3rd year, though we've been experimenting with using partial fallow and using field peas as a green manure for a portion of that fallow period. Another important part of the first grant is to extend our results to farmers, stakeholders, lifelong learners, and institutionalize organic farming principles in resident instruction. And one of our newest projects is down at the bottom, and you can see that right here. We have just started a student-run organic demonstration farm on UNL campus. So that's going to be really exciting. We've got a bunch of the students participating. We're going to have a cover crop experiment out there. So we have a PhD student. He'll be doing a cover crop experiment. We're just starting out with a 1/2 acre. And then we're hoping to renovate a hoop house and develop some small vegetable plots. So that's going to be a really nice thing for our campus to have. We also have a series of NEB Guides in Nebraska, and we have 4 NEB Guides right now. 3 are being under review. We hope to complete 10 NEB Guides, trying to give farmers in our area the basics in organic production, but sort of tailor it to what the actual conditions are in Nebraska. Another project we have that was part of our grant was to look at flaming for weed control. Dr. Knenzovic and Dr. Gogos have developed this service manual. You can go online and have access to it. Thanks to the Propane Education and Research Council, they were able to publish this, and it's a project. I've been working with 4 organic farmers in our area, testing out the development of this flamer to use on corn and soybeans primarily. However, we're finding other uses with that, so that's been another benefit of the project. And flaming might seem, like a great tool if you have particularly wet weather, but as you know, we haven't been having the wettest weather, so I think it has a lot of potential for drier weather too, to help farmers not do so many rototowings and different cultivations. So we see some promise with his work there. Part of our OREI grant is to look at on-farm research for organic production, and we have been working with several of the organic farmers in our state. I think organic farmers, or like most farmers, are experimenting all the time, but we're trying to get them to replicate and randomize their experiment. So these are some of our projects that we're doing there, and they're pretty varied, as you can see.
Unmuted.
That's it for me, and I'm going to hand this over to my colleague, Charles Shapiro. Thank you.
Okay, Charles, you should have the screen control, so just click on the screen once to activate it, and then you can move it with your arrows. There you go.
Okay, thank you, Alice, and thank you, Liz, for that introduction.
Muted.
In addition to Stephen Knezovic, who's a colleague of mine up here and working on the flaming, we've had many support from organic farmers around the area who've given their advice over the years, as well as technicians. So they do a lot of the work for us, so I wanted to make sure they got thanked. As Liz said, in Nebraska we have a lot of range of conditions agroecologically. They say it's more varied from the western part of our state to the eastern part of our state in terms of rainfall and some other climate factors. So in terms of managing nutrients, especially release from organic matter, we can't really generalize over the whole state. What I'm going to talk about today is the research we're doing in the northeast corner of Nebraska, where the Heskel Ag Lab is, on that dryland organic farm that we have within our research station. On our ground, it's sloping, maybe 6% to 10%. The fields, as you can see, are kind of long rectangles. Each one is only a few acres in size, from 3 to 5. Makes it a challenge to conduct replicated trials because of the constraints of terraces, but we try to fit them in as best we can. There are 2 main projects that we're doing up here at the Heskel Ag Lab. One of them is the one I'm going to talk about today, the first one on the screen. We have had this grant with winter wheat, and our part of it here is to help create a cropping system in eastern Nebraska that includes more than just corn and soybeans. And so adding a small grain, preferably a winter small grain, was what our objective was. The winter wheat would be taken off in mid-July, and so there'd be a fairly long period from mid-July until the next spring when a row crop could be planted. So our goal was to try to manage that resource and put something productive in there, hopefully something good for the soil erosion, and then either adding organic matter or nitrogen or both. And so that's kind of the emphasis of where we were going with that. In addition, we've also been working on understanding the nutrient-weed interactions in organic row crop systems, but I won't have time to discuss that this afternoon. So the main thing we want to look at in our winter wheat system is that cover crop establishment, and then how to synchronize nitrogen release for both yield and protein. And Richard Little will talk more about the wheat quality, so I'm not going to spend too much time on that. in front of you, you have a fairly complicated schematic of our thinking on this, and I'm going to walk through this so you can understand where our research is coming from. We split it up into two sections, nitrogen timing and the cover crop and its management section. But we're looking at a rotation, and we can start anywhere in this cycle. But if we have a row crop or an alfalfa, we split the two between a crop that would be using the nitrogen up in the soil versus the alfalfa that might be putting some nitrogen back in. We have an opportunity for manure spreading. If we have a diversified smaller farm, they would have livestock of some sort, and they might have an opportunity to spread manure either on the alfalfa before it was tilled in or after corn harvest. We would plant our winter wheat, and then in an effort to use a small amount of nitrogen but maybe influence quality, we chose to apply foliar nitrogen later in the season at jointing time of, I think, where we tried to get it on in the winter wheat. And then we would take the winter wheat off, and then we had another experiment where we would go into wheat stubble with either grass or legume cover crops, depending on whether we were going to end up with a grass-based row crop or a legume base. We wanted to keep kind of a rotation of grass, legumes, and we would be researching both the growth of these cover crops as well as how we were going to kill the cover crop or manage that residue to get into the corn. So it takes a little bit of timing and planning to set up these experiments, because we have to have alfalfa, then a year corn and alfalfa, the winter wheat, and move on to the legumes. So it takes a fair amount of coordination to get that done. What I'm going to talk about is mostly very briefly, I'm not going to spend any time on the cover crop species or how much they produced or any of that. I'm going to just briefly go over what we found and how the termination method. And termination methods, we had the flaming, as you could kind of see in the background. We used some of the earlier prototypes that Dr. Knezovic was working on, and then we had conventional disking and the roller crimper that Liz mentioned. One point that I do want to make is that that period between winter wheat harvest and the fall freeze, the rainfall is very variable in Nebraska. We could have perfect conditions to get the cover crop up and growing, or we could have a 6-week dry period and a very poor stands. And the 2 years that we did the cover crop study, 2009 and 10, we had one of each. The first year was tremendously dry, and we had a better stand of volunteer winter wheat than we did of the cover crops. And then the second year we had a fantastic stand, and this is the good stand that I'm showing you with the hairy vetch in front of you. So that's really critical, whether we get enough residue to make it worth the effort, and that's part of the problem of being on the edge of the corn belt. When you boil everything down to what we did in terms of the termination methods, I have put together the 2 years data, 2009, 2010, and where we disc, which would be the normal termination method, or where we grow cover crops that would winter kill. In this case, we had either buckwheat, Sudan grass, or soybeans, or oats. This would be more of a normal dryland yield for that kind of cropping system. In addition to the benefit of the disking, it also allowed us to plant earlier. So we had reduced yields where we either flamed or used the roller crimper. And part of that problem was, you see here, the reduced populations growing into the cover. And then there was also a weed control factor. So both getting a good population and then consistently getting the weeds killed were the two problems we had. I failed to make a similar slide to this for the soybeans, but the soybean yields were actually a much higher percentage of the disc or winter kill, and we did better trying to manage the grasses before the soybeans. And so on average there was a 36% reduction in the soybean yield for either one of the roller crimper or the flamer kill methods, whereas we had about a 68% reduction here on the corn. As Liz mentioned, we are doing more work on the roller crimper. We think we can make that work more effectively. This experiment was our first effort, so we're not saying that we're done with that part of our investigations. Now to move on to the nitrogen component of our work, you may recall we would either have alfalfa or corn as a previous crop. Then we applied manure. We actually applied manure either in the fall or in the spring, and we applied it at 3 rates, so 0, 20 tons, and 40 tons per acre. In the spring application, it was early in the spring, and it was on established winter wheat. And then we had 3 rates of foliar N, 0, 10, and 20 pounds applied later in the season. And this attempt here was to improve protein more than to improve yield. We did struggle to get OMRI list approved products to do this, and we used different products each year due to the certification issues. An aerial shot of our organic farm. You can see in 2008 where we had planted the corn. We have blocks of corn, and then within each of these blocks we put strips of the manure and then substrips of the foliar N. And this is it in more detail. We repeated the study at a different place on the farm in 2010 and 11, so we have 2 cycles of this experiment. Again, due to time limitations, I'm not going to give a lot of detail. I'm going to go right to the yields and what we found. At the end of the first cycle, one thing we found was that our first attempt at undercutting that alfalfa with the Noble blade was not entirely successful, and we did have a fair amount of alfalfa that did not get killed. It survived and competed for moisture with the winter wheat, and that certainly affected yields following the alfalfa. A side benefit of that was after we took the wheat off, the alfalfa came back, and we didn't measure it, but we probably had about 1.5 tons of alfalfa in September that we could have harvested for use as a forage. Our best treatment was fall applied manure at the 20-ton rate without any of the foliar N, and we had about a 12 bushel increase compared to where we did not have any manure applied at all. We did have this problem that's listed there where we got on the fields a little bit too late in the winter, and there was damage to the wheat with the manure application. So unless you can get on there fairly early and not damage the wheat, that's probably not a good time to apply the manure. We didn't really affect protein at all, and that's because it was fairly high even in our check plots. So where they've had the problem was where protein was much lower than 13%. 2011, we had similar response. We did a better job of killing the alfalfa. We had actually much poorer stands coming out of the winter because we had poor stands coming in. It was very dry, and we actually abandoned one replication because of stand. But the bottom line was that the lower rate of manure on the corn did pretty well compared to nothing. And with the alfalfa, we did get a bump with the 20 tons of manure. In the second year, we were on a site that probably was phosphorus deficient as much as nitrogen deficient, so probably some of the alfalfa response here was to the P, not to the N. So just kind of to review what we found here, went over briefly, was that we did a pretty good job with a corn, manure, winter wheat. We did see no evidence to use any foliar N application. And then the choice here, probably if we're going to go with the roller crimper, and in soybeans would be the way to go with a grass cover crop and follow it down here, as compared to moving this around and ending up with corn. So to finish off, our big desire is to be able to consistently get good cover crop growth, but that's really mediated by rainfall in the August period, and in this part of the state we need to expect that we'd have some failures in some years. We're working on the crimping. We kind of have similar responses where it's easier to kill the grass than the legume, and that's shown up in our work. In terms of influencing the winter wheat, we don't have a real big fertility problem on our soils. They're high native fertility, so we got a marginal increase with the smaller rate of the manure, and found that the spring manure, even though it might have more nitrogen available early, we got more damage from the plant damage than benefit. And we found no reason to go with a foliar N late in the season. So with that, I'd like to hand control over to Richard, who will talk about his work with wheat.
Thank you, Charles.
Unmuted.
Hi, I'm Richard Little. I coordinate the organic small grains breeding research, and thank you, Charles, for a good lead-in with the wheat research. We go a little farther than just working with one variety, as in Charles's research, and find that there are differences among the varieties in responses. Our current project, funded by Ceres Organic Trust, is a follow-up to the OREI project on breeding small grains for organic systems. Bay State Milling and Heartland Mill have contributed substantially through quality testing and milling. Artisan baker members of the Bread Bakers Guild of America are graciously volunteering time to do baking tests this year.
Muted.
The empty table provides an outline for how our story ends, with 19 promising experimental hard winter wheat lines and varieties remaining out of 55 total tested over 4 years in our variety trials at 4 locations. We also tested 400 experimental lines at 2 locations each year that fed into the variety trial nursery. As we shall see, most of these promising lines have major drawbacks. The story also evolves from taking notes on a dozen traits, including canopy cover and rust diseases, to finally focusing on those that really matter, shown in the row across the top, including coleoptile length, seed-borne diseases, fusarium head blight, and whole wheat quality. Yield has almost become a secondary concern. Before I tell this story, I will give some background on hard wheat and adapted varieties. Because our small grains breeding program is in the plains, we are devoted to breeding hard winter wheat, which predominantly goes to the bread market. Other pockets outside of the Great Plains with significant hard winter wheat acreage are indicated in the brown ovals. The adaptation of wheat lines developed at UNL is shown by the black circles. We also know that UNL and SDSU varieties are adapted to New England states based on recent testing there. The 1,200 crosses that we make each year have enough diversity to be adapted to the larger area represented by the red circles and beyond, likely beyond. For more background, I refer listeners to an eOrganic webinar on winter wheat breeding basics. Next, I will talk about key traits and how our emphasis changed over time. Originally, we focused on the functionality of white flour for making bread. When our advisors asked us to strive for nutrient density in our research, our focus shifted to whole grain flour, since the wheat germ has higher levels of vitamins. With our new focus on whole wheat, we recognized that we needed to be more concerned about vomitoxin from fusarium infection of seeds, since it is concentrated in the bran layer. We also recognized that processing may be more important than genetics for retaining or releasing nutrients. For instance, 2/3 of ash content, the minerals, is in the bran, but it is not bioavailable unless it is released from its bonds to phytate, which is also present in wheat flour. It is possible to breed for lower levels of phytate and to increase levels of iron and zinc, which is a current project of USDA ARS at Lincoln. However, there is a quicker way to make the minerals available. Disable the phytate by fermenting with sourdough. Hence our recent focus on baking sourdough bread. The low pH conditions of sourdough have several favorable consequences for nutrient bioavailability and for improving bread volume of whole wheat bread. Low pH favors enzymes that deactivate phytate, favor other enzymes that release the main phenolic antioxidant in wheat, the ferulic acid, from its bonds with arabinoxylan in bran, and enable other enzymes in some sourdough cultures to break down the razor-like bran fragments which puncture dough and keep bread from rising. Before we focus on sourdough, I was selecting only the strongest gluten lines to compensate for the effects of bran. This goal may no longer be valid with sourdough. Therefore, we've asked 8 artisan bakers to bake loaves from whole ground flour by Heartland Mill using a standardized sourdough method developed by Tom Leonard. For later reference, notice the 2 protein contents for the cultivar being tested, in this case McGill. The loaves shown here are the early results from Jim Williams of Seven Stars Bakery in Rhode Island. To introduce you to our breeding challenges for other traits, I want to first describe a key challenge facing the organic wheat industry. Notice the high concentration of pink dots in the panhandle, which indicates many acres of wheat production. This is the same map with an overlay of organic farms represented with black dots. Notice the small number of organic farms in comparison to the large wheat acreage. Think of how much more wheat is available to commercial millers of conventional flour than to organic millers. Think from a miller's perspective how much more difficult it is to blend wheat to certain specifications when there are so few sources. Now consider that 3 varieties predominate on organic farms across Nebraska. 70% of wheat sourced by Bay State Milling in the area near the Wyoming border is buckskin. Carl 92 dominates organic farms in southeastern and south central Nebraska because most millers seek at least 12% grain protein. In 2009, Carl 92 at 13% protein was 0.5% higher or more than most other varieties and was the only line that met Bay State Milling's criteria for baking quality among the 20 lines they tested for us. Arapahoe is the 3rd popular variety among organic farmers in Nebraska. In recent years, the only wheat varieties offered by a Nebraska organic certified seed business were Carl 92 and Arapahoe. The limited number of varieties and the small volume of organic wheat production make each year and its hazards a risk for organic millers. From the farmer's perspective, even though they would prefer higher yielding varieties, it would take a lot of convincing for them to change from a variety that they know the millers will accept. When we started testing varieties on an organic farm in 2006, Wahoo had surprisingly good yield performance during dry years compared to conventional plots. Since then, we found that only 30% of the highest yielding lines in conventional trials were also the highest yielding in organic trials. We also found that buckskin and Carl 92 were among the lower yielding lines in our trials. What traits in these lines did farmers value more than yield? We already noted that protein content for bread quality was important in choosing Carl 92. An important trait for farmers in the panhandle is coleoptile length. A longer coleoptile is needed when planting deep to reach moisture. Buckskin has a 4-inch coleoptile. In our F6 nursery, only 11 of 280 of our experimental lines have long coleoptiles as long as buckskin, which severely limits our ability to select high-performing lines with this trait. In our advanced nurseries, the number has dwindled from 17 a couple of years ago to only 1 of 120 lines this year. But despite the rarity, we have an exceptional long coleoptile experimental line, NEO 8457, that also has higher than average protein content. Organic grain buyers informed us that the wheat they buy must have a test weight of at least 58 pounds per bushel. This poses a dilemma for us because some of our best lines for yield in organic systems that also have good quality, namely Wahoo and NEO 3490, are inherently low in test weight. We need to either abandon these good lines or to educate the value chain to accept low test weights for specific varieties. Fusarium head blight is a menace to grain quality in its production of vomitoxin, especially for the whole flour market. Our experience with screening for FHB is that we need several years of data to be confident in labeling a line as resistant, because it is difficult to get good infection rates and spread of the disease some years. There are no real resistant varieties, only varied degrees of susceptibility. The most consistent, least susceptible varieties are Lyman and Expedition. Overland and Goodstreak are often mentioned as resistant or least susceptible lines from UNL, but I'm not entirely sure that Overland fits that category. Two seed-borne fungal diseases show up when fungicidal seed treatments are not used as in organic farming. Black tip, shown at the top, and common bunt, shown at the bottom, discolor flower, and common bunt imparts a fishy odor. We expected great things from Wesley and Rubidoux varieties based on high yield and excellent bread quality in conventional trials. Our hopes were dashed by common bunt that reduced their yields to the bottom of the organic trials in 2008. This disease is difficult to screen in a breeding program because incidence is sporadic. So we ignored common bunt for a few years until in 2012 at Clay Center, conditions converged, late planting into cool soils, that enabled spores to infect the seedlings. 3 of our most promising experimental lines, including Carl 92, had severe common bunt. We also had high expectations for excellent bread quality and yield for our most recent release, McGill. But in organic plots, McGill had both high yield and low protein, and the baking results were equally poor. Surprisingly, our highest yielding varieties, Overland and McGill, had the highest levels of total phenolic antioxidants across locations over a 3-year period. On the other hand, one of our lowest yielding lines, buckskin, was also the lowest in antioxidants. This inverse relationship between yield and antioxidants was not as evident for our other lines. As for Overland, it is suitable for the breakfast cereal market, but unfortunately makes poor bread. Now we make our way back to the protein content traits so favored by millers and farmers. We wonder if high protein lines might deplete soil nitrogen that is already scarce on many organic farms. Perhaps it is possible that a line with low protein content but higher in quality protein would use less nitrogen. Preliminary studies indicate that 3 of our most promising lines, I won't give the names of them here, bake well at grain protein content as low as 10.5%. As noted earlier, promising cultivars are in the process of being evaluated for quality at multiple protein levels. The purpose is to determine the protein threshold at which they bake well. In summary, although quite a few lines yield well in organic environments that do not necessarily perform well in conventional management systems, very few lines have adequate resistance to seed-borne disease and FHB. Very few lines have long coleoptils. Very few have both excellent bread quality and yield. To view the data in this table, a NEB guide will soon be available on selecting winter wheat cultivars for organic production. I conclude that focusing on yield and white flour bread quality did discover several adequate lines for organic production that mostly overlap with recommended lines for conventional production, but they did little to improve the chances of attaining lines with the optimal combination of traits to match the needs of organic markets and production within specific ecozones. To improve our chances for success, I recommend that before testing experimental lines in replicated organic yield trials, selected lines should be screened in a low nitrogen environment using an assay that can predict whole wheat bread quality. The lines selected for screening in this nursery should have exceptional resistance to seed-borne diseases and FHB for subhuman ecozones and should have long coleoptils for semi-arid ecozones. And I'll turn this over to Vicky to talk on the antioxidants. Thank you.
Unmuted.
Ready? I am online.
Okay.
And let's see how far I'll stay. I have disconnected a few times inadvertently. So I'm going to need assistance from people at the WebCare session to make sure my slides
presentations
keep on going with what I'm saying and that sort of thing. But
okay, that's fine.
I did hear,
We'll just tell us when to advance the slides and we'll do it.
okay, this sounds great. I did see everybody's talk from my iPad, so I was able to bypass my PC and just go to my iPad. So I just don't know how to run my presentation from the iPad. So we'll do that from the conventional rounds here. So to get started, do we have these in PDF file or are they in the regular PowerPoint?
Well, right now they're in PowerPoint, and we've also uploaded a PDF file to the registration page. But right now we're showing the PowerPoint.
Okay, that's great. Okay. All right, so if we go to our front page, this is just basically introduction of what that I do and what I participate in this group, and that I study the organic crops grown in Nebraska. And I specifically emphasize those components in organic crops based on the phenolic-based antioxidants. And the crops that I'm interested in are the soybeans, the grains, the corn, and the high oil sunflower seeds. I'm going to turn the slide next. And this shows an outline of what I'd like to present today. What are phenolic compounds first and foremost? What are goals and our outcomes from this research? And then to show you an example of some of that research that we are doing, and the data results from that one study, and then ongoing studies that are continuing through these studies, and some of our final impacts. So what are the phenols in the plant-based antioxidants? In general information, phytochemicals are those that are widely distributed through the nature. They usually are found in plants, but not necessarily so. You can find them in fish, you can find them in butterflies, certain insects, that sort of thing too. But they're most notably known for their presence in plants. Now, when they are present in plants, they're present in very small amounts. We're talking parts per billion, parts per million. They really are so much smaller than our main components of food. But what's really interesting about these types of chemicals is that they're very chemically diverse compounds. And we have found that consumption of foods rich in these chemical diverse compounds, or what we call micronutrients at this stage, is that we have been shown that they have been lower links to risk for cancer, heart disease, arthritis, Alzheimer's, diabetes, and Parkinson's disease. Amongst of them, there's others, of course, to talk about here, but I just named a few. So what a phenol is? A phenol is anything that has this ring structure, a six-faceted ring structure with bonds conjugated throughout the ring structure, and an OH group sticking from it. It then can morph into a higher structure, again with some more acetic type compounds coming out of them. I wish I could have a pencil to show you these. But you can see from the example I've given you, still see the hydroxyl groups with the ring structures and some of the different types of stereoisomers that can arise from this. These are called phenolic acids, and that they contain all the COHO groups. And then we got the stilphenoids to the right. The stilphenoids have two phenolic rings, and the stilphenoid I put up here because it's called resveratrol. Resveratrol has been implemented in so many different types of health mechanisms and health care products that I didn't think we should go without talking about these just a little bit. Oops, going the wrong way. Sorry about that. And then on top here, we've got some other polyphenols that group by, coming together more closely. They have some interesting qualities that most people don't study too often, but I do want to tell everybody that they still are in the plant system, and we don't know how they act synergistically with other plants to elicit a higher health benefit than what we would see if they were gone. And then we got my favorite, the flavonoids. The flavonoids are characterized by these three-winged structures, cyclic structures. You've got two close by each other that are bonded to one another, and another one that has a long chain and then is bonded further away. The one closest to the middle one is called A. The one further away from the middle is called B, and the one in the middle is called C. So when we test all of these compounds, we have a method in place that we can test all of them, and we call those total phenols. The total phenol test. When we go to flavonoids, we can further disintegrate them, deconstruct them into their individual structures. So here we got an isoflavone that goes into isoflavone, that goes into genistein, that goes into adenosine, by just virtues of little changes in these functional groups. Now, isoflavonoid is known for being in soy. You have, if you go down to the flanose group, they are also shown by the fact that they have these double bond O group. But then it goes over to quercetin, and quercetin is a ubiquitous type of flavonoid that's found in most foods. So there's quite a few of these types of flavonoids, plus they can be conjugated to so many different types of sugars and, glyco components, things like that, that we can get up to 4,000 to 8,000 different types of phenols. So to monitor all of these, we call them the total flavonoids. So what we want to do is that antioxidants or phenols are called secondary metabolites synthesized by different plants in response to stress. That is what they do. They try to suspend the plant from any type of wounding, stage of growth, drought, weed, insect pressure, UV radiation, soil nutrient content, location, infection, and cultivar. They are not considered the main component of foods at all. They are contained within the food, again, at very small levels, but the small amount present gives a lot for their pack. So what we wanted to do was determine the phenol flavonoid levels of organic grown groups in response to different factors, considering that they would be going through these different types of stresses all the time. And so our outcome was to provide information on the optimal crops, cultivars, and organic farming practices to implement in different areas throughout Nebraska that promote some healthy crops that are prevalent in our state. Here's a picture of Nebraska with the various organic lands that have been set aside by the university for us to do these types of experimentation. Charles has talked about Heskel lab, which is shown at the northeastern side of the state. Then we have this Ag Research Development Center, which is Meade, and then we have the Central Ag Lab Clay Center, which is about the middle south central part of the state, and then over by Sydney, the High Plains Ag Lab. And we have organic plates or organic plots in each of these areas now. So one of our first studies is that we wanted to determine how phenols and flavonoids could be affected when grown through these different areas in our state, but in regions that are very different in terms of their agricultural, So we had 19 different wheat lines grown under organic conditions. We grew them in 4 locations throughout Nebraska, and then we monitored for their total phenol flavonoid levels, and then we monitored them for their antioxidant capacity. We are in the process of doing peroxamates, which includes protein, carbohydrates, water, minerals, let's see, fat, and carbohydrates there too. And our long-term related back to yield, and we want to relate this back to yield and other quality characteristics that our colleagues are also working on. So just to show you a couple things, because we have so much going on, I wanted to show you the differences in total phenols in terms of what happens when they were planted in Heskel Lab, Ag Lab, the Ag Research Development Center in Mead, and South Central Ag Lab and Clay Center. And if you look at the top of Heskel Lab, you can see that there are differences in the amount of phenols being produced in response to the different lines of wheat that were used. The most predominant phenol was by millennium. The least amount of phenols was overland. However, what I thought was interesting about Heskel Lab is that even though you saw these high and lows, the overall variability between each of these lines were very low, which, you know, very low variability is a good thing. And our average between all of those types of lines were 0.389 mg per gram. That's not the highest we've ever gotten from Heskel. We have gotten up from Heskel to be about 0.5, 0.6 grams per mg. Milligrams per gram. That was in 2008. So, you know, but we had a lot more variability then too. So what we have to determine is variability worse or getting higher yields with higher variability a better option to go to. For the Ag Research Development Center, we found that the highest yields were NEO 5425. Again, it wasn't that much higher, but we got about 0.4 mg per gram for this phenol. But the rest remained about fairly the same, not a lot of variability between them, and together as a group we obtained about 0.382 mg per gram of phenols. And for the last one, clay counter, this was the lowest in terms of producing phenolics. It did show several trends, and that a lot of the lines show various trends, with the highest trend being overland, with the lowest trend being buckseed. But again, I was happy to see that the variability was more consistent than what we have seen in the past, even though in the past we have gotten to be about 0.6. So, but that it could go back and forth as well. Now for the flavonoids, and flavonoids, because they are known through health promoting research that flavonoids are probably a little bit more potent than the flavonoic acids, things like that, the other things that I've shown you, even though they are around at a lower amount. So for the Heskel lab, there was no statistical difference between any of those lines during the wheat lines during 2011. They all showed about 0.1 plus or minus 0.02. For their overall amounts to be about, average amount to be 0.099. We did see a little bit more higher averages with the the mead, and that was 0.166. But again, the variability, although it doesn't look like it compared to the Heskel, was actually a lot less than what we saw in 2008. But the yields were about the same. The yields don't change with the flavonoids too much throughout the years. So, and we see about the same McGill, NE05, all of those being the higher producers. Going back now to Clay County, again, you see not a lot of variability between the flavonoids. Some of it, the NE093 show high yields, the Darrell shows lower yields, for overall average of 0.0988. So not much different than Heskel labs. And if you look to the right too, I have the average antioxidant capacities, and they were about 440 micromole per 100 grams of product, compared to the Heskel, which was 4,008 micromole per 100 grams, which was a lot higher than what we saw with the mead, even though it seemed that they might have a little bit more phenols there. And after looking at all of this data, I am really getting more and more convinced it's just not total that we're looking at. And the total thing that we're looking at is that what we need to do more compositional analysis of each of these to determine what types of flavonoids or phenolic acids are really exerting their effect. So we do have data from 2008 to 2011 to look at that type of data, and we are going through each of them methodically to really understand the genetics versus the environmental have on each of these wheat lines at the different types of the different locations in Nebraska. But other work that we are doing that I don't want to count out, and because of lack of time, is that we are evaluating organic grown crops for phenolic compounds and their health promoting the health promoting benefits, I should say, across cross types. We do know that sunflower seeds produce much higher phenolic and flavonoids and antioxidant capacity than wheat does. And we do know that soy does through certain time too. We just don't know how they are affected by cultivars, by locations, by nutrient treatment across years, disease stressors. And when I say disease stressors, I'm not only talking about the diseases that the crop itself can face, but I'm also talking about the disease stressors that humans eat and we also can assimilate into our own bodies. And we also have not done any work with conventional crops. We're talking to a few people about looking into this, but this is really a major area that we need to address to understand conventional crop farming practices with organic crop farming practices and how all they all help produce a better, healthier food. And so the final long-term impact of my research, which I hope will integrate to everybody else's research, is to produce a healthy crop, be it from what we eat, but how we grow them and how it affects the environment and so forth. So with that, I thank you all.
Unmuted. Okay, Liz, we'll hand things back to you. We don't hear you yet, Liz, so I think you're...
Yes.
There you go. Okay.
Thanks, Alice. I think we're going to be listening to Jim Brandle now.
Oh, okay, sorry. Yeah, it's Jim's turn. I'm sorry about that, Jim. Let me switch the control to Jim. Okay, there you go, Jim. Just click on the screen there. Okay.
Okay, great. Well, let's go back a slide because there's some folks we want to mention. As Charles mentioned, there are often lots of people involved with these studies that are never mentioned. And three, in our case, Amy Oden, Heidi Adams, and Nancy Beecher were all graduate students that worked on this project over time. So they are as important to us getting this data together as the major investigators. Basically, we know that extinction is a natural process. We know we've lost species over time. The fossil record is there. We are approximately 1,000 times higher rate of losing species now than we were then. And as we look to the future, we can expect that rate of loss to continue. The question then is, can we, or with our practices, sustainable practices or organic systems, contribute more to the protection of biodiversity? And more importantly, can we measure these impacts and help the organic farmers invest their dollars wisely in these conservation practices? Whoop. It is slow, isn't it? This is results from one of the early studies that made some comparisons between bird abundance and richness on organic and non-organic fields. And one caveat to this study, while the organic fields and the non-organic or conventional fields, they had the same amount of biodiversity in vegetation surrounding. In other words, they had the same vegetative structure in adjoining fields or non-crop areas and so forth. So the differences we're seeing are attributable solely to the practices, or we think solely to the practices that are associated with organic systems. So this is quite positive. We followed this up most recently as part of the overall study that Liz explained when we started off, and started looking at the biodiversity associated with three different scales, or landscape scales. One, the local field scale, then a smaller one called the field scale, which we don't report here. Local scale is basically the farm itself. The landscape scale is everything out to about 5,000 meters, which is about 3 miles. So a swath around the organic farm in 3 miles in diameter. If we look at the graph on the left, we see that some species respond, and look at the bottom, percent of block grasslands, these are alfalfa, these are large patches of grass or alfalfa. We see that the grassland sparrows responded well to more and more grassland being available on a farm. Similarly, the dick thistle also responded well or positively to more and more alfalfa. So these two species are encouraged by having larger tracts of grass or alfalfa. In contrast, the horned lark responded negatively to increases either in alfalfa or grassland. And so what that says is it likes more diversity in its habitat, more non-crop areas that may not be dominated by grass or alfalfa. On the other side of the image, there is a landscape scale graph that goes out to 5,000 meters, so percent of grassland within that surrounding area. And here we see that the western meadowlark and the ring-necked pheasants both respond to more at the landscape scale. So an individual farmer activities on a particular field or on a particular farm don't necessarily have a positive influence on these two species. Collectively, people in the area, farmers in the area, have a very big response to these or impact on these species. And here's some other shrubland birds. These tend to be species that like more diverse habitat. They don't require the large open areas of grassland that the other species we were talking about. So you can see a number of different species, and I won't go over these, but these are all showing positive responses look at the percent grass cover and the local diversity, or Shannon index, which is just a measure of how different types of vegetation in the area, how different the non-crop areas are. And so both of those respond fairly well to those kind of things. And if we go back and look at the diversity up here in the aerial shot, this is one of the farms. You can see the diversity, especially in the farm itself. Different fields, different grassland areas, different vegetation types. That's what we're talking about. So when we're really looking at is, while farmers are interested and always like to know what birds are out there, they want to know what the benefits are to them of having these extra birds out here. How do I manage my ecosystem? How do I create conditions that promote the biodiversity, either planned biodiversity, which is your crop rotation, or the associated biodiversity that's in those other areas that we don't farm. And so these then are basis for what we call ecosystem services. And these are the things that the ecosystem provides to us. And they can be broken down in a number of different categories, and we'll get to that in a minute. But ecosystem services include things like clean water, clean air that we take for granted, areas of riparian buffers that clean up water that runs off a field, crop borders that clean up runoff. These are all the kinds of things we're talking about. So what we started with were characteristics or criteria that are important to estimating what we might consider to be what is a healthy farm, if you will. We use bird diversity because that's something we have a lot of data on. And we use crop yield since this is really the basis of economic return to the producer. We went to him from those two starting points. We went out and talked to a number of all of our cooperators on the organic system and looked for and asked them for suggestions as to what else they think is important. And there we are. So crop richness might be something that they would identify for us. Livestock richness, speaking how many different types of livestock do they have, how many different crops do they have. Both of these deal with their rotation and production systems. From ecosystem service standpoint of view, we were looking at how much of the farm is in crop and how much is in non-crop areas, how much is in borders, how much is in riparian systems, and so forth, or windbreaks. We looked at ecosystem richness, and on the off chance that they had a rare or endangered species, we wanted to know that. None of our producers had any of those rare or endangered species. On ecosystem services, we can break that down into three categories. Provisioning, which is basically what we produce, the crop yield, and marketing opportunities. Where can I market that crop? What kind of variety do I have? Do I have options in terms of marketing? Regulating are things like soil and water conservation. And the cultural things, well, how satisfied are you with your operation? Does it give you a good life? How long have you been at it? These are the criteria then that we collected from information we collected from the producers. And so then with that, we were able to rate different farms in a biodiversity score and an ecosystem service score. And here are two organic farms. The one on the left has more diversity, an index value of 90, versus the one on the right with an index value of 75. And looking at those two aerial photos, you can see the difference in the landscape vegetation types that are out there, the crops that are out there, the different conditions of the field. The one on the right has several large fields that are fairly uniform, homogeneous. The one on the left has a lot more differences or heterogeneity in it, and thus the different index values. We also rated one of those. We all talked about the organic side at the Agriculture Research and Development Center. And this is one of the sections that's located there. This is the agroforestry section. The organic system that people have been talking about is this center one right through here. And so we rated it along with its surrounding habitat. And again, we came up with index values that were much higher in most cases. The biodiversity index was about the same where it fell down is that there are no livestock on this particular research area. When we look at the ecosystem services, it fell down because of the market opportunities. The university markets under a very rigid system and can't always look for the best opportunities. Basically, we take it to the local elevator. In conclusion, basically, things of value to us is habitat diversity. This is the thing that came across loud and clear. The more diversity you have in your habitat, the healthier that ecosystem is going to be. These are three other systems that we looked at for one of our companion studies. These are all field windbreaks that looked at how many or how far away from a particular habitat, in this case a riparian system, in these two cases windbreak systems along the edge of a field, how far away will birds forage into that field.
Muted.
And so here we look at the responses of the avian foraging sites. In other words, how far out can we expect birds to be predators on various crop insects? And so you can see quite a difference. These are ones that feed within 20 meters of the edge, and these are all fairly shy birds, ones that don't really care for people. Some of the other ones are much more ferocious, if you will, and much more daring, is maybe a little bit too personalized. But if you look at some of these, there are some of the species, the blue jay, the oriole, the robin, kingbird in particular, forage just about anywhere, and they're relatively unaffected by us, if you will, and so they will forage farther out into the fields. The kingbird in particular is one that really has a lot of promise as a predator for various crop pests. John Quinn is now a faculty member at Furnham University, and really should be the one
Unmuted.
this talk. He did most of this work, and his dissertation that came together for this particular thing. He's got some contact information there and a couple of key papers for those who really want to follow up on the details, because we certainly gave you more of the general highlights and not much of the real gory details. So with that, I think we turn it back to Liz, who's going to sum it up. Is that right, Liz?
That's right, Jim. Thank you so much. That was great. Can you hear me? Okay.
Yep.
So I just want to summarize that part of the success of our research is developing partnerships with several of the organizations that are found in our state. And we also had a strong relationship with the agricultural farmers around each research center, and we had a very good organic farmer advisory committee that provided a lot of good guidance on
Muted.
for some of these research projects that would actually be beneficial to them and really address some of their production needs. I'm trying to, there you go. The Organic State Advisory Committee came up with a vision statement for our group, and it basically talks about some of the strategies that we are going to be developing for transitioning from conventional agriculture to organic whole farming systems, working with established farms as well as new farms so they're ecologically self-renewing, socially responsible, and profitable, and that they'll provide nutrient-dense foods and ecosystem services and education to current and future generations, which you hopefully have heard running throughout the other talks today. I just want to point out that there are approximately 47 people that have worked with the Organic Working Group, researchers, technicians, graduate students. So it gives you an idea of the scope of people who have been able to participate in this research program. And we're at the end. I want to thank you for your attention, and everyone's talks are very good. And if you have any
Unmuted.
questions, please ask them. And I would also point you to our website. Right now it needs to be updated, but you can definitely go there and get contact information, or you can certainly contact me and I'll help you find anybody that you need to for information. Thank you. Alice?
Okay, thank you. We're about to begin some time for questions and answers. And okay, yeah, there's a question about whether or not you track where GM alfalfa is planted in Nebraska, if someone could address that.
Well, I'm unaware of that we're doing something like that. I don't know if anybody else would know.
Okay, yeah, don't forget to unmute your microphone if you wanted to speak up there.
This is Charles. I don't know either.
Okay. All right. Well, we don't have any other questions. Wait, here's another one. Okay. Were there interactions on the phenol flavonoids, or were results consistent between years? That's a question for Vicki. Were there it says, were there year interactions on the phenol and flavonoid?
Yes, there actually is. We're most definitely seeing that across, especially like between 2008 and as we're going into 2010 and 2011. What is interesting is we want to now integrate some of the environmental aspects that were going on at those times as well.
Okay. We have another question about whether millet is grown in Nebraska.
Millet?
Millet, yeah.
Yeah, millet is grown in Nebraska. This is Liz. And we are actually going to be experimenting with millet at the Heskel Ag Lab this year. Typically, you'll have more millet grown in the western part of the state.